In today's lecture, we're gonna discuss about the three different types of calibration methods, and for most of you it's gonna be a reminder. Now remember, we said last time the purpose of the calibration is so that you can relate the signal from the instrument, and so that you can relate it to the concentration, which is what you're interested as an analyst. So there are three types of calibration methods, the easiest of which is external calibration method. Now the external calibration method, is where you prepare the standards, and of course the standards, you're preparing them with very known concentrations, so you're very accurately determining, the concentration of the standards, and I hope it's clear, standards have got known concentrations of the analytes, and then you're analyzing them using the instrument separately, from the unknown, which is the sample. And after that, you would generate, your calibration, from the standards. And the calibration, of course, has got, the regression equation, meaning it's a linear relationship, that is a signal, that is a concentration. And then after that, you run the sample separately from the standards. You're going to get the signal from the instrument. And then based on your calibration, you can determine the concentration of your sample. Now take note of this. Yes, the standards were ran separately from the sample. And so one huge disadvantage of the external calibration method is that it does not account, for matrices that are present in the sample, because the standards are pure. They've been ran separately from the sample. So you made an assumption that the instrument is gonna respond in the same way to the standard as well as to the sample, which is not completely true, because we know the sample has got matrices, and matrices can interfere with the analyte detection. And so to solve the problem of matrix interferences, you have to use a different type of calibration method. And the calibration method that you can use to solve for matrix effect is what we're going to talk about, standardization calibration. So just to summarize very quickly, with the external calibration method, as I've said, the errors involved is that huge assumption that the instrument responds to the sample in the same proportion as to the standards, which is not completely true because, again, of the presence of the matrices. So the way you correct, for these errors using external standard calibration is using blanks. And blank, remember, is a solution that you run on the instrument, but it doesn't have the analyte in it, but possibly it has got every other matrix, that is present in the sample, but doesn't have an analyte in it, and that is what you refer to as an ideal blank. However, it's very difficult to get an ideal blank. It's almost impossible, really, to create an ideal blank. Take an example. You're trying to analyze, say, lead in a coal tar, that you obtained from Northwest Territories. Ideally, if at all you can get an ideal blank, the coal tar, should have everything. Rather, the ideal blank, should have everything in the coal tar other than lead analyte that you're analyzing, which is totally, of course, impractical. In fact, in an ideal environment, you should ensure that you expose the blank to the same environment as a sample. So that means you have to take this blank, all the way, and expose it to the same environment as a sample, which is, again, is impractical. So for that reason, ideal blanks, are impractical, in analytical chemistry. The closest we get, to a good blank is using the so-called reagent blank. And the reagent blank is where you've got the solvent, plus all the other, reagents that you use to prepare the sample. So that one you can easily do in the lab. But again, sometimes that takes quite some bit of time to prepare. And so you would find that the most efficient route, especially if at all you don't have a very complex environment, is where you just simply use a solvent blank, meaning you just take the solvent that was used to dissolve the sample and you use that as your background. But of course, it's not an ideal situation because it won't correct, for the matrix effects. And then after that, you would subtract the signal from the blank and you subtract it from the samples as well as the standards. And that way, you correct, for external standard errors. But as I've said, what is very difficult to correct in the absence of an ideal blank, would be a sample that has got matrix effects, which again is not very easy to correct without an ideal blank. Because remember also, lots of things can happen to the analyte, as you're doing the extraction and so forth. It can undergo different types of chemical changes, due to the exposure to the environment. So in an ideal situation, as I've said, that should be accounted for in an ideal blank, but that is essentially impossible to do. So in practice, the solution to matrix effect, the practical solution to matrix effect is what you refer to as, the standard addition method. And before I get to the standard addition method to clearly explain what it is, maybe this slide is a good one to explain to you, the severity of the instrument response, when you've got matrices. Because what happens is, you've got matrix interferences that suppress the signal. And so that's why you can see, if at all I've got matrix effects, my calibration is suppressed compared to if I do not have matrix effects. So matrix effects, are very, very significant and have to be accounted for or dealt with. And as I've said, the solution to matrix effects, is standard addition calibration method. Now, what is standard addition calibration method? So the standard addition calibration method, it's where you analyze, the standard in presence, of the unknown. And in practice, the way you do this is you take, say volumetric flasks or containers, in the lab, and then you would pipette it, with a volume of a known volume of the unknown, and you pipette them in equal volume for all the, the volumetric flasks. And then after that, you would spike a known concentration of the standard in increasing volume in each one of these volumetric flasks. And hopefully after that, you would make it up to volume, using a solvent. And so you can see in the very end, the standards have got, the unknowns, meaning the matrices are all the same, and they've got increasing concentration, of the standard because you spiked them, with a concentration of the standard, but with different volumes. So for example, in vial 1, you can spike it with 0 microliters of the standard. In vial 2, you can spike it with 5 microliters of the standard. In vial 3, you can spike it, with 10 microliters of the standard and so forth. And so in the end, you've got standards that have got analytes and samples and the matrices, all in solution. So you can see, that all of them have got the same matrices. And then after that, you would analyze them on the instrument. And then you can generate, a linear calibration. Now, in this case, the y-axis remains the signal from the instrument. So, for example, if at all you're doing AAS, like you're gonna do this week, the signal in this case is gonna be absorbance units. All right. And then in the x-axis, in the case of a standard addition, you would plot the volume of the standard that you added in each volumetric flask. For example, like I said before, initially I spiked 0 microliters, maybe 5 microliters, 10 microliters, 15 microliters, and so on. So again, the x-axis, you would plot, the volume of the standard, that you added in each volumetric flask. And of course, after that, you would get a linear calibration. So how do you then determine the concentration of the unknown? Remember, the concentration of the unknown is sort of present in each of these volumetric flasks. So the way you do this is using this equation here. The unknown is going to be equals to the intercept, and then you multiply that by the concentration of the standard, and then you divide it by the volume of the unknown, and whatever you get is going to be in the concentration of the unknown. So how do you get the intercept? Of course, you already have the equation of the line, y is equals to mx plus b. If you get y is equals to 0, that is the intercept. And the x in that case then is going to be b over m, and of course it's going to be negative, but you only take the magnitude of it. You only take the magnitude of it, and you do not take into account, the negative, the negative sign here. All right, so it's going to be negative b over m, and then you multiply by the concentration of the standard, and then you divide it by the volume of the unknown, which is the same in every volumetric flasks, and the result that you get is going to be the concentration of the unknown. So fairly straightforward to determine the concentration of the unknown in using standard addition method. Now, the other calibration method is a so-called internal standard method. Take note of the following. Standard addition corrects for matrix effects. However, we still have another problem. Instruments in general, are known to have some random variations over time. So they may have, drift over time. Another thing is when you're doing analytical chemistry, chances are you took the sample and then you took it through a couple of steps in terms of sample preparation. Now we know when you're doing sample preparation in the lab, chances are, there are some random errors also, that are present, when you're doing sample preparation. There may be losses of the sample, probably due to evaporation. There may be isomerization taking place, and so forth. And as I've said, also remember, instruments in general, have got, random errors, due to different factors. I'll give you an example. If at all you're doing gas chromatography, typically you inject very small concentrations, normally something like 0.1, to 1 microliters. And of course, you're supposed, to inject it very reproducibly from standard to sample to blank and so forth. It's very difficult, to reproduce very small amounts of sample injections. And so you're going to have quite a bit of errors, associated with injections when you're injecting very small amounts of volumes. And that, of course, affects, your calibration as well. And so to correct for sample preparation errors and to correct for random errors or variabilities of the instrument, you typically use the so-called internal standard calibration method. And the way it works is you take an internal standard, and then you would, an internal standard meaning the concentration is known, and then you would spike it in the constant amount in both the samples, the standards, as well as the blanks. So for example, if at all you're analyzing, say, caffeine in green tea, you would take an internal standard and you would spike it in known concentration in all the tea samples, as well as all the standards and all the blanks. And then after that, of course, you would analyze it using the instrument, to generate, the calibration. So the only difference really in terms of the mechanics of it, between internal standard and the external method is that in the y-axis, instead of just plotting the signal, in this case, you plot the ratio of the internal standard divided by the ratio, divided by the intensity, of the standard. All right. So you take the internal standard signal and then you divide it, by the signal, of the regular standards. And the x-axis, is gonna be simply the concentration, of the standards. Okay. In the next slide, I'm gonna differentiate between two things that I know can be confusing: the standards and the internal standard. I'm gonna try to differentiate the two, so that it's fairly clear. But again, the y-axis, you take the internal standard signal, divided by the signal of the standards. So how do you choose the internal standard? So the internal standard, should be a chemical that you choose that is structurally similar, so very, very close to your standard or to the analyte that you're analyzing, but it should be distinguishable. So it should be very, very close in structure, but it should be distinguishable, from your analyte. Now, the internal standard, the other characteristic is that it should respond to the detector in the same way as the analyte. Another important thing, it should of course be stable. The internal standard should not interfere, or react, with the analyte that you're looking for. And of course, simple things, like of course it should be soluble in the solvent that you're using for the analyte or the standard in this case. So you would find then a good internal standard would typically, on an ideal internal standard, is typically the isotopic label of the analyte. Now, isotopic labels really mean, if at all I've got, say, a benzene ring or a benzene compound such as that, typically internal standards, would be, of course, this is carbon 12, carbon 12 and so on. Would be, I enrich some of these carbon atoms and I will replace them, with carbon 13. So maybe I would take these three and I would enrich them, to create carbon 13. And so in the end, I'm gonna have a heavy benzene, all right? Meaning, of course, the molecular weight of the heavy benzene, is gonna be the, the internal standard. So it's very similar, to what you guys do in the NMR, where you use deuterated solvents, and deuterated solvents really are nothing but an enriched, isotope of whatever compound, that you are looking for. Meaning, if at all I was, talking about, chloroform, for example, a deuterated chloroform would be where I replace this hydrogen with a deuterium, through an isotopic enrichment. And there are ways of doing that. And it's a very common process, for example, in cancer, diagnosis, this process of enrichment. Okay, so an ideal standard, therefore, ideally would be an isotopic label, of the analyte, which, as I've said, is often generated through isotopic enrichment, of that particular analyte. Now, the problem is isotopic enrichment process is fairly expensive. And so you've got most of the compounds out there, you wouldn't be able to get an isotopic label for those compounds. And so it's very, very rare sometimes, to get isotopic label of any analyte that you're looking for. And so you have to look at another alternative in the event you don't have an isotopic label of your standard, which is the ideal internal standard. Okay? And the next best option of an internal standard, would be things like an isomer. You can decide to use, a structural isomer, say of your analyte. That's another acceptable, internal standard in the event that you cannot afford or it's not available to buy an isotopically labeled compound as your internal standard. So I hope it's fairly clear. And again, as I've shown you, the y-axis, it would be the signal of the internal standard divided, by the signal of the regular standard. And the x-axis is just simply the concentration of the standards. And this chromatogram shows you something very important, that the internal standard here should be distinguishable, from your other standard, which is the analyte in that case. Those of you who did Chem 211, are familiar with this relationship, which explains the idea of the 1-point standard addition, 1-point calibration, for internal standard method. So I hope it's clear to you how you do calibrations using the 3 different methods, namely, the external calibration method, the standard addition method, and the internal standard method. And I'd like you also to be able to know why you use each one of those methods so that you can determine, depending on the problem, what's the ideal, method of calibration that you should use. And so you've got a dry lab assignment to be able to practice, all those different types of data manipulation using the 3 different calibration methods. And this is very important, because all your labs are gonna be based on calibrations. And so I want every student, to be very, very comfortable, with data manipulation, using different types of, calibration methods. So I've already posted the dry lab for you on Blackboard. It's got, I guess, 4 different questions using different calibration methods. And so you're supposed, to do the calculations and, submit the report back on the 3rd of February, which will account, for your first lab report. To make life easier for you, especially for some of you who are not familiar, with Excel as a software, I've posted for you a calibration template. And the calibration template is supposed to help you, do, your dry assignment, your dry lab assignment, as well as all your other labs. Okay? And it's got, say the standard addition, calibration method, it's got, the external standard calibration method. And remember, as I've told you, the internal standard method is exactly the same as the external calibration method. The only difference is you just need to take, the signal, of the standard, divided, by the signal of your internal standard, and that is what you plot, in the y-axis, and the x-axis remains exactly the same. So you'll have, of course, to take the template and modify it, for the external calibration, and create, another sheet, for determining the internal standard calibration, because it's exactly the same. The only difference is that. So I'd like you guys, to be able to use a template. Of course, you have to modify it appropriately. I've only given it to you as a start, so that, you're not too lost, in terms of doing this. Ideally, I shouldn't be giving you, of course, the template, but hopefully, it's gonna help you guys a bit, to solve some of these problems. So in essence, if at all you're doing, say, standard addition, all you're gonna need to do is just replace this data set, with the data set, in question or in the regular lab as well. Make sure you understand, what all these equations mean, so say for the least squares method, that's the equations you use, for the standard for the standard deviation, of the unknown, that's what equation that you use. So be able, of course, to manipulate, your data, based on, the template by changing, of course, the variables as needed. N, of course, refers to the number of data points. Of course, you have to keep changing that depending on the problem in question. All right. And of course, so be able to understand what's going on in the template. And for those of you who did Chem 211, I think it's fairly straightforward because, you've already done these before. And so you'll be able to find the template, in the dry lab folder, And you can use again the template, to complete, your dry lab assignment, which is posted for you. And as I've said, it's due February the 3rd. So until now, what I've talked to you is about using calibration, and you generate the equation of the line y is equals to mx plus b, but the signal, we've only been taking it as one single variable. All right, so I've only been doing the so-called univariate calibration. So until now, what I've shown you is univariate, calibration. I've assumed the instrument is only giving me one single signal. However, you're going to realize, depending on the type of the instrumentation that you are using, sometimes, you would get multiple signals. I'll give you an example. Assume you're analyzing caffeine using FTIR. The caffeine IR spectra, looks like that. So the question for you is, which instrument signal should I be using? If at all I'm doing univariate calibration, chances are, I may come here and just use, the signal that has got the largest intensity. But sometimes you get interferences happening in that region. So what's the other option? So the other option, would be to use the other types of signals. However, an ideal situation, would be where you use not one signal, but instead you use multiple signals to build your regression model. And that is what you refer to as multiple linear regression. That is very, very common if at all you are dealing with very complex samples. And also it's very, very common, when you're trying to build a fairly robust calibration in an environment, that is fairly complex. I'll give you another example. If you had a mixture such as this, you've got now caffeine, you've got theobromine, you've got theophylline, and possibly they are all present, maybe in a cough syrup, and you're trying to do quantitative analysis. Now, what do? As you can see, all your IR spectra very much overlap. In fact, if you put them side by side, you might not be able to tell a clear difference, between all the three different compounds. And so meaning your univariate calibration, may not work, especially if at all you're at only FTIR in the lab and you need to do quantitative analysis. So what's the solution? Again, the solution is to do multivariate regression, where you use, all these different types of signal to build a calibration model. Maybe another example, just to show you what I've just said, if overlap, the three compounds or the three spectra, you can see I can't really tell the difference between them. So they're totally indistinguishable, meaning, again, I have to use a statistical tool, to do multivariate regression so that I can actually differentiate, between which signal I should be using to build my model. Maybe another example is this. And so the way you do this in terms of building the regression models for these very complex mixtures where the signals are totally indistinguishable, would be you create standards, say of the three different compounds, theophylline, theophylline, caffeine, and theobromine. And so you've got different set of standards. Say standard 1, I mix it in proportions of 1 to 1 to 1. Standard 2, I mix it in different proportions, and so on. And then after that, I run on the FTIR, and I would get, different, types of signals. And then based on these standards that are in a mixture, I can use statistical tools. To build a multivariate calibration. And so using the statistical tools, I'll be able to use matrices, that help me determine which are the most optimal signals I should be using to help me build the most linear model. And there are many statistical tools out there. Excel is used quite a lot. It's not very robust. There are other things, like SPSS. And if it was not for for this pandemic, in previous years, students in Chem 311 have used SPSS, to play with this kind of data set, to build multivariate regressions and so on. There are other softwares that you can use like R, and so forth, to build, again, multivariate regression. But again, in this course, again, I won't go into great detail about multivariate regressions. But I would hope for some of you interested, especially for your research projects, we can play, with multivariate regressions, so that you're familiar with it. Now take note of this. Sometimes we call multivariate regression in analytical chemistry as chemometrics. And it's actually the basis, of artificial intelligence, or what you refer to as data analytics, which is a very, very common thing, in our age now, which is how do you get, the most useful information out of very, very complex data, like I've tried to show you, in the previous slide. And of course, most of this is not as complicated. They can get, a lot more complicated. So let me just very quickly, highlight for you, different types of multivariate calibration platforms, that are typically used. Like I said, you're already familiar, with the univariate calibration, where you're just using a single variable, a single signal. Multivariate linear regression, again, that's where you're using multiple types of signals, to build, your regression model. Now, in some cases, when you've got very, very many signals, you have to use statistical tools, to reduce the data to only give you variables that best predict the concentration of the unknown. And again, things like SPSS and even Excel, can actually help you to do that reduction method of data. Now, there are other tools that people can use. Again, there are statistical packages present in SPSS, like, principal component analysis, partial least squares, and many, many other types of, regression models, that you can use, which again is beyond the scope of, the Ken 311. But again, they're fairly simple tools, actually, that are fairly usable. So let me very quickly, go through 3 case studies or 4, where chemometrics or multivariate regression, is very useful in, okay? Case number 1, which is fairly straightforward, so you're trying to generate a calibration of a single compound, but a single compound is being analyzed using, say, chromatography or using spectroscopy, meaning you get multiple types of wavelengths or multiple signals. So instead of just using one signal, you can use multiple signals, so that you can build a robust linear regression model. But again, you can see it's fairly straightforward. In this case, I just have a single compound and it's giving me multiple signals. Very similar, to the example that I gave you with the FTIR and caffeine, where you've got multiple signals. And so instead of just using one signal, you would use multiple signals, to give you the linear regression model. A more complex case, is where you've got, say, 4 compounds. I've given you an example before, 4 compounds, and you're trying, to build a linear regression model for the 4 compounds, but they are already in a mixture. But again, I don't want to do chromatography. Maybe I just want to do analysis of the 4 compounds simply using spectroscopy. And so based on that, I can again analyze them in different proportions, make standards in different proportions, and use statistical tools like SPSS, Excel, and so on, to build the best regression model, for each of the compound in the mixture. So very, very useful, if at all I don't want to do chromatography, which takes a lot of time, and very useful also if at all I'm trying to detect any impurities, say in pharmaceutical formulations. Another more complex problem here now is where I'm trying to analyze, say, polyaromatic hydrocarbons, which are carcinogenic compounds, present in a coal tar. Now, you can see, unlike my previous problem, where I've got only 4 pharmaceutical compounds, in this case, I could have, up to 20 or more different polyaromatic hydrocarbons present in my coal tar, meaning it will be very, very difficult for me, to actually build a very accurate standards because I do not know what else could be present in the coal tar. And it's impractical, of course, to try and generate, all the compounds that are present in the coal tar and make standards, of all the compounds that are present. And so again, the only thing that I can possibly do is get as many compounds as I can, build as many standards as I can, analyze them using the instrument, and again, use a statistical tool, to try and build, the best regression model, using the compounds, that I have. And that's what sometimes people call training the artificial intelligence or training the AI, which largely refers, to making as many standards as possible, so that I can use them, to build, my regression model. And again, you can use that using common statistical packages such as SPSS, and so forth. Now, the last case, is this, where, you are trying to not necessarily determine the concentration of the unknowns, but in this case, you're just trying to group different sample specimens and trying to see, if they relate with each other. Sometimes people call this, discriminant, discriminant analysis, and those of you in statistics, I'm sure you've come across it. I'll give you an example. Say I'm trying to analyze, whether wheat grown in Saskatchewan has got the same protein content as wheat, that is grown, say in Alberta, compared, say, to wheat, that is grown in another tropical destination. So I'm trying to analyze the amount of proteins that could be potentially present in different, in wheat grown in different geographical regions. So in essence, I just want to cluster, the data, to show that, there are relationships between wheat, that is grown between different, geographical locations. So I'm just classifying the data. For example, I could as well try to analyze, say, the quality of wines. Are they very similar? If at all they are grown between two different or three or four more different geographical locations. So again, I'm using statistical softwares, to do discriminant analysis. Or even another excellent example, some of you are interested in environmental analytics. You go to the ocean and you find, there has been an oil spill, and the analytical chemist, you've been asked to determine which company's vessel or ship actually spilled the oil. And so again, you're not really doing quantitative analysis. All you're trying to determine is, whether the oil that you find in the ocean, in an oil spill, very closely matches, say, the oil from Syncrude or the oil from Mobil company, or the oil from, other companies as well, like Shell and so forth. So you're just looking, for clusters of compounds that are present, that's going to help you show there is a correlation, between, between the compounds. And so it's generally used, again, for discriminant analysis and not so much, to do quantitative analysis at all. And again, chemometrics is used a lot, again, a statistical approach, to doing data analytics. It's used a lot, for dealing with these kinds of problems. So you can see chemometrics is used for a variety, of different kinds of data analytics. And I should mention this, really, it's the future, as you well know, about artificial intelligence being a very important, part of, analytics, not only in clinical diagnostics, but in all sorts of different types of fields. I'll give you a case study. I've previously given this to my previous students, say on the problem of PAHs, that you are discussing. I guess question, case number 3. So the way you would do it, I'm trying to analyze, say, PAHs in coal tar. So what I would do is I would build, say, 25 different standards, say, of pyrene, acetylnaphthalene, anthracene, and so on. All these are different types of PAHs. I make different types of concentrations. So because you can see these are standards. I run all these standards of known concentrations. You can see that I've ran them on the instrument and they've given me, this type of data set, the next type of data set corresponding to those concentrations. And so based on that, I know the concentration, I know the signals, I can use something like SPSS, to build a model like that tells me, which are the best wavelengths that best predict each of those types of PAHs, and so I can build 10 models, for all these different types of PAHs. So not very difficult to do. Again, you can use statistical packages to do that. But in this course, we won't do that, because of time. But I just try to explain it, that it's a very common, data analytics tool, that you can use for multivariate regression. So it's not just univariate regression, you can also do multivariate regression, for complex data, like I've just tried to explain.